Great. So welcome back, everyone. It's a great pleasure for me to introduce Justin, who will present some background about the MS2LDA tool. And afterwards, I will also talk a bit about MolNetEnhancer. And yeah, I think, Justin, the floor is all yours. Can you hear us, Justin?
Yeah, I can hear you. Can you hear me again?
Yes.
Cool. Okay. Well, thanks, Madeleine, and it's my pleasure to be with you, rather through the internet. But I hope you can all hear me well. So the aim of this blog is to get you introduced into substructure mining and also some chemical class annotation. And so I will start with some background, and then for the more hands-on part, Madeleine will take over. So basically, it's everything around getting more out of your untargeted mass spectrometry data, right? And so let me. Oops. So with my group in Wageningen, we apply and we develop tools to do that in the application areas of natural product mining and discovery, and also the food metabolome. But of course, these tools are more widely applicable also for the DOM example that you have this week, and also environmental sciences and other areas where metabolomics, small molecules play a role. So if you're interested in our work, then we have a website where you can see which research lines we have, and also keep an eye out for open positions if you're looking for jobs, because there some positions will be opening up in the course of the next academic year. So untargeted metabolomics as a whole is on the rise, I think, because of several reasons. Some of them may have been already discussed with you, or you have seen yourself, but instrumentation got faster. We've got better generic extraction protocols, and also automation plays a big role there. And you can see it also in publications, right? More than 1,000 publications were published last year with untargeted metabolomics mentioned. However, I also strongly believe that it's supported by computational metabolomics, because as you will have seen, you can generate now a lot of data, MS data, MS data, but how to analyze it. So, and there all the computational tools that came out over the last years, including MZ-Mine, molecular networking, and others, have played a pivotal role to drive and targeted metabolomics. But now the question is how to annotate substructures or structures based on MS spectra. So if there's one takeaway from this blog, then it's this. If you look at this apartment in Copenhagen, so basically this was the view of out of my window when I lived in during the time when I lived in Copenhagen, and I liked the building, but as you can see, we can quickly recognize it's a house, it's a building, because we can see windows, we can see particular structures like the roof. And other ornaments, and that's basically when we do pattern mining on this object, we can quickly recognize it's a flat, right? And maybe for those of you that live in Copenhagen, you will recognize even that it's from there, based on some characteristic features. And it's this aspect that we use over and over again in different omics fields, including metabolomics. Because with pattern mining, we can group, we can start to annotate and connect things. So the current format is not so interactive, and some of you have seen this picture before, I'm sure. It's when I took it close to San Diego, and our human brain within a few seconds will recognize typically two groups, right? There's two different groups of animals, and luckily both of them are not so dangerous to us, especially on a distance. So, and the idea behind pattern mining and using it in metabolomics is that you can exploit the grouping to propagate structural features, right? So if one bird can fly, then, well, maybe all the birds can fly, okay? And there's two examples of molecular families, molecular networking, and you will have seen that in this week already, how you can use that. And now today we will focus a bit more on extracting the building blocks of metabolomics, or the substructures, because also that we can use to group and propagate features and annotate whole families. So, well, in short, how does it work? So the tool was coined in 2016 as MS2LDA. So the name is like going from MS, mass spectrometry, to LDA, using the latent dirichlet allocation, topic modeling. Well, let's quickly forget that again. We just focus on how it works. And also MS2 is like using MS2 data with LDA. So it's important to mention this. It's unsupervised, untargeted substructure discovery. You don't need to know what is in your sample to try to find the patterns that could be linked to particular substructures. So we're going from text to molecules, so inspired by text mining. So for example, you have different documents with words, and you want to quickly analyze that to see what they're about. Then we can try to recognize words that are often co-occurring in text, because they may represent a particular topic. And as you can see on the left-hand side, we have different documents, and the words are colored by their topic. Okay, and I hope you agree that the red words represent football-related topic, and the blue words could represent a business-related topic, and then finally the green words represent a more environment or energy-related topic. And already a few things pop up, like, hey, we can recognize different topics in one document, and we can recognize the same topic in different documents. The blue topic present in the two documents. And if we think about substructures and molecules, it's exactly what we want, right? So we have molecules, they can consist of multiple substructures, but also the same substructure could be reused in different molecules. So bang on, yeah? So let's see, how do we go to the molecular world? Well, the documents become the molecules, the spectra, and the words become the fragments or the neutral losses. And then finally, you can see that the word club on the left upper corner is mentioned like 4 times. And typically we can do a word count in text mining, and that indicates the importance of a word for a topic. Well, on our end, in the mass spec world, we don't have words, but we have the spectra, and we can use the normalized intensities, basic intensities, as a proxy for this word count or the importance for a particular topic. And if you think about it, the higher the normalized intensity, also the more likely we will actually see the peak in mass spectrum occurring under various conditions. Okay, so back in 2016, we analyzed beer, and I saw some pictures already from leading up to the workshop. So I'm sure that you have seen some beer, and so we measured some Scottish ales. And in there, I could annotate based on the patterns and using databases already quite some biochemically relevant substructures, amino acids, aromatic substructures, sugars, etc., etc. And we also did validation using 5,000 GMPS spectra and 2,000 mass spec spectra. And I want to let you know, so 5,000, that was all there was back in 2015 when we started that process. And nowadays, 7 years later, I think we are nearing the 500,000 annotated spectra in GMPS, and of roughly 22,000 unique molecules. So I'll give you a few seconds to think about that. And I will come back to it at the end of this short primer. So actually, we managed with the data that was to validate quite a couple of the substructure patterns that were found unsupervised. So in this case, the peaks are related to adenine. And you can see that even though the spectra are quite different, we were still able to connect them. And also, we could identify the green loss that was related to the ribose part of the molecule in the middle. And together, the two parts form the complete molecule of adenosine. So we could even use it to dereplicate one molecule completely. So of course, annotation is difficult. So and we should not make our lives more difficult than needed. So can we make this process more effective? So yes, and efficient. So we call it MotiveDB a bit later, which is a database of always detectable master motive substructures. So basically, you put in spectra, you get out a motive like this, which I got from bacterial data. I analyzed the data. I found out that this particular peaks were related to the lactone ring of actinomycin D. And now it is part of this annotated motive set in MotiveDB. If you have bacterial data, which you suspect that they may be related to Streptomyces or Salmonella bacteria, then it makes sense to add this motive set, this annotated motive set, because if related molecules are there, the annotated motives will be linked and it will be quicker to replicate your sample. And the nice thing is you can add both annotated motive sets, but also still add free motives to do the unsupervised substructure discovery in your own samples. So, okay, it's all nice, Justin, but you know, does it really work, and can you really use it on experimental data? So, so I brought a small plant sample from the Rhamnoceae plant family. To cut the long story short, we have two different plates, and we wanted to know, can we recognize differential chemistry? Yeah. For today, I will not go into too much detail on that question, but there is a paper published back in 2019 by Madeleine, who will follow me up, and Ki-Ben Kang, who is now in Korea leading this research group. And so if you want to know more about that, please have a look at the paper. So I mainly talked about substructures so far. There's also this chemical class component of workflows. So for that, it's important to realize that over the last 4 or 5 years, more and more ontologies are being coined and used. We have classifier, we have empty classifier, and that help us to annotate structures in a logical way, in coherent way. So it's important to realize that by the use of these ontologies, all these chemical class annotations are related to each other. Why? Because we can recognize more broad terms like lipids and lipid-like molecules in the green bit in the middle of the lower figure, but also we can go more down in terms of specificity to, for example, 3-terpenoids. So 3-terpenoids are part of these lipids and lipids-like molecules, but they are more specific annotations. So, and then MolNetEnhancer, that was published 3 years ago now by Madeleine et al. That brought it all together, at least the many of the tools that were out back then, into one, at the end, Cytoscape network, where you can then see all the results on top of each other and combine the annotations into one hopefully coherent picture, right? So basically, you can follow the workflow stream going with your samples. So in this case, plant-based samples, but they could be from different origins. And then you do the multiple mining and library matching and GPS, molecular networking and MS2LDA, substructure discovery. Then you can add annotations from tools like dereplicator, FarQuest, MolDiscovery, and then you can apply network annotation propagation that Madeleine will also highlight a little bit, I think. And then we can run the chemical class annotations using all those annotations to get a high-level overview in the right bottom corner, but also we can focus on the more chemical details when we combine the substructure annotations. So the reference in the plant journal is the work on the Rhamnoceae data that kind of was the lead up to the MolNetEnhancer workflow. So did it work? Well, so this is the molecular network of the Rhamnoceae plant family, and each color represents a different chemical class. So, and we have a long list. So, but for those of you that work with plant data, there may be, I mean, if I were in the room, I would ask you to raise your hand, but I can't do that now. So you can raise your hand to yourself. But I hope that you and also others will recognize that there's many plant-related classifications in the list, right? Flavonoid-related stuff, phenolic glycosides, and triterpenoids. So that all kind of seems to work quite well for this example, at least. So the automated annotations based on chemical class can help you to start to get a feeling for what's in your data. So, and then the deeper insights can occur when you map the motifs on top. So, and then you can see an example here of a flavonoid trioglycoside family based on the chemical class annotations. But then if we look at the motifs, the map motifs, you can see a clear difference between the top and the bottom part. And then actually it turns out that there's one subfamily based on temporal and one subfamily based on the flavonoid quercetin, two often occurring flavonoids in plant material. And then another example is from the triterpenoids. Remember the lipid-like kind of class of molecules, and they are often conjugated in plants. And here we can see that this whole family contains of triterpenoids, but we can recognize protocatagoic acid, phenolic acid, and coumaric acid as conjugation agents for these different triterpenoids very quickly now. So here you can see how bringing it together can help you to more quickly identify or get yourself, pointing out to the relevant or interesting parts even of a family. So I thought, okay, now you've got the concept of pattern mining and then inspiration by text mining. So let me also very briefly point you out that in my group we are continuing this line of research. And again, you can see here that in text mining we have different sentences where we can recognize if words mean the same thing or have a similar meaning. And if you're not from Italy, then I think you agree that those two sentences are roughly meaning the same thing. If you're from Italy, I'm sorry, cappuccino and coffee are both contain coffee, right? So, and on the metabolomic side, we can do the same thing, right? We can also try to identify relationships between fragments. So the word becomes the fragment or the loss again, and the sentence becomes the whole spectrum. And we can then try to run similar algorithms as they do in text mining. There it's called word2vec, to see if we can establish relationships. And we can then create new embeddings, new dimensions. And here, as an example, I created this, right? Two new ways of looking at the data, one positive affection, one hot drinks, and then we can score each word for how well it kind of fits into that new embedding, new way of looking at the data. And for example, the word coffee is completely gives a 1 in hot drinks and a 0 in positive affection, but the word likes and loves, they are the opposite, right? So of course, normally you learn this stuff from a lot of text, or in our case, you learn this kind of relationships from a lot of spectra, and then you get values. And in the end, we can then use this novel mass spectral embeddings to compare spectra again. So, and I brought one example. So we can now use on that concept, we can do fast and scalable analog search with Spectrovec. So only a few seconds, we can mine a large library of spectra for structure-related molecules. You can see two examples on the right-hand side for a cyclic peptide and a lipid-like molecule, where we indeed find structurally very related molecules in the database. And the good news is that if you're interested in this, check out the recent preprint by my group and Nick de Jonge on MS2 Query that uses Spectrovec, MS2 Deep Score, and other recent developments to come up with one new machine learning based way of doing library search and analog search. So check it out. Yeah, I'm getting to the end of this primer. So I promise you to come back to this mass spectra. So basically, all these tools that you heard about and many others are really depending on annotated data to be able to develop and test and validate, right? So if you are working on your project and you get a chance to share your data, your annotations, your metadata with the scientific community, do it, right? Share your smiles, both in real life and from the molecules you annotate, and share your spectra, right? And my group and other groups will be really happy, right? And thankful to you. So and just because I think it's important to show that all this chemical stuff can be very intriguing and maybe sexy as well, is that I'm also working on ways of how to bring, let's say, this mass spectral embeddings that I talked about in the last few slides, more to make them more understandable to students and also to fellow researchers. And this is an example of how a visualization team at the Wageningen University called Wonder, they developed this visualization tool, this 3D tool, to browse the embedding. And basically each dot you see here is a spectrum from a mass spectral library from GMPS, colored according to their chemical classes that we discussed. And basically the students had the chance to browse through this and then, for example, go to a particular place in the embedding and check, hey, here I see that most dots are white. So basically the embedding was able to group structurally similar molecules in one place. And then, of course, it's interesting to start to find out what kind of molecules are they, and are they indeed structurally related, and how are they related? Are they maybe measured also on the same instrument, etc., etc. So if you're interested in all of this and more, then and it went a bit quick, which I can imagine, then there is a good review, I think, but I'm a bit biased because I'm one of the authors. On all of this and more. So on substructure annotation, class annotation, and network analysis, and really how we can go to large-scale substructure-based analysis. So yeah, please, the reference is also on the slide. And with that, I would like to thank you for your attention. If you're interested in my work and in my group's work and in pictures of Wageningen and the environment, then follow me and or my group on Twitter. And I'm happy to be in touch with you if you have additional questions. So I will stick around till the end of this block. So, well, thanks a lot. And the floor is yours, Marlijn. I think I will stop sharing.
Thanks a lot, Justin, for this really inspiring talk. It's really impressive how many tools you are developing. Unfortunately, we won't be able to show all of them hands-on at the workshop, but we will be looking into MS2LDA job submission through GNPS in a minute. But first of all, are there any questions to Justin's talk? From here, live in Tübingen, or maybe from the chat as well? I will have to look.
I just wanted to add that I uploaded the slides to your Google Drive on the Wednesday, so in case you want to look back at them.
Really great. That's really appreciated. I think there's one question.
We just talked about series, but he also talked about substructures, linking to fragments. But I guess to me, at first view, it sounds like you're doing something similar, but using a totally different kind of algorithm behind it. Is that right? But if I have data, would you suggest try both because one will have the advantages here, the other one the advantages there, or are there special use cases where you say rather use this one or rather use that case, depending on what you kind of data you have?
Right. I will just quickly repeat the question for you, Justin. I will summarize. I hope that's okay. So we had just a talk from Kai about serious CSI finger ID, where Kai also talked about substructures. So the question is, how does MS2LDA compare to serious CSI finger ID? And would you suggest, if you have your own data set, trying both, or what's the advantage of one versus the other? I can quickly add, like, I will a bit go into that as well when I will introduce MolNetEnhancer.
Yeah, so, well, very brief answer will be that if you have the time, then for sure try both and see what kind of information you can get out. And when you talk about substructures, I mean, it's not really well defined. So, what I think, so the patterns that MS2LDA picks up can be anything from a carboxylic acid group to a amino acid to a larger scaffold like camphor or quercetin. And the series finger ID workflow, of course, it will also annotate the molecules. It works with fingerprints as well, and that fingerprints can also represent particular functional groups or substructures. So I mean, the approach is different, but in the end, the information you can get out will partly the same. Yeah. So I think the an advantage of the MS2LDA motive DB workflow could be that you can pick out the relevant motive sets that have annotated substructures that you recognize as being relevant, so you can quickly screen your data for them. But of course, serious finger ID has the advantage that it is the state-of-the-art workflow to get to elemental formulas for most of your metabolites, especially the smaller ones, and also state-of-the-art in terms of the correct candidate. So if you can use that information also as part of MolNetEnhancer, then I would certainly do that.
Perfect. Thanks, Justin. Any more questions right now?
And Kai, if you are around there, then feel free to add or to counteract.
So I think naked work between both methods is MS2LDA is AXPROWISE and CSI is PROWISE. So AXPROWISE methods are trained on all the data outside, and PROWISE methods are trained basically on spectral libraries. And I think the consequence of that might be that over there, MS2LDA is really nice for inspecting the data, visualizing the data with all the networks, because motives might correspond to really large patterns. Or like, I mean, we show a lot of examples. And I think the CSI patterns are often more smaller functional groups and stuff, which are well defined in spectral libraries, which we can learn from spectral libraries. My master motives would be also stuff which is not contained in spectral libraries, but maybe unique to a data set, and which then shows you, okay, these 10 compounds from a data set share something in common, and that's probably something can only get from MS2LDA.
Thanks, Kai. I'm not sure if the online participants could hear, but I'm sure you can contact Kai later on as well if you want more in-depth explanation. So I think I will continue with showing you how to do the job submission of MS2LDA online on GNPS. Justin, I know you have to run for another meeting, but just feel free to leave any time you have to go, and if you're still here, feel free to chime in anytime if I'm doing any mistakes or showing something wrong. Just please, yes, shout out. But yes, so you can run MS2LDA from within GNPS, and so how to do this is in fact really straightforward. So I just have to get rid of this thing. Okay. Well, I'm not sure in whose account I'm in, but it should work. I can't log out because of the tab. But so basically, if you go, you can either go to the feature-based molecular networking job you submitted yesterday of your own DOME lab, interlab study data, or you can also use the link that's provided in the slides in the Wednesday session. We just select the job here. And basically what you will do is you go down here where it says analyze with MS2LDA. So if you click on this.
Sorry.
Yes. Oh, okay. Yes, of course. Sorry, I forgot that. Is this working now? Okay, great. Okay, so we are here. GNPS automatically redirected us to the MS2LDA job submission. So the first thing you can do is you can give your analysis a title here. It's really just a title of your choice. So we can say DOME Interlab study and then MS2LDA. And here GNPS will automatically populate everything you need. So you don't need to do anything here. Just to explain, GNPS will put the MGF file that you produced within MZMine, and then also the CSV quantification file. And here you just specify that your output format is derived from MZMine preprocessing. Then there are some specific MS2LDA parameters you need to set. Usually the defaults are just fine. So the bin width is instrument dependent. So if you have Q-exactive data, which I think is mostly the case, right? Correct me if I'm wrong, Daniel. For the interlab studies are mostly Q-exactive, so you can leave that at the 0.005 bin width. Then the number of LDA iterations, and just how many times LDA will iterate. So it's 1000. The default is also fine. Then the minimum MS2 intensity, 100 is also a good value for Q-exactive data. If you have tough data, you could consider to go down to 100. And this one is basically the most important parameter, the LDA-free motifs parameter. So here you define how many motifs you expect in your data sets. And this again is extremely data set dependent. So you might have to try out different settings until you get a good number of motifs. But the data sets I usually work with, I always will estimate around 200 to 300 motifs. But it really depends also how rich your spectra are, so how many peaks you find in your spectrum. And then Justin introduced, there is also the motif database. So this is basically what you define down here. So there is different motif databases where you have annotated motifs. And here you would, of course, select the motif databases that you would intuitively expect in your data set. So we have here a Euphorbia and Rhamnacea plant motif databases, which of course we don't intuitively would expect in environmental samples, so in DOME samples. So I would exclude those for sure. And also Streptomyces and Selenospores. I'm not a microbiologist, but I think I wouldn't expect them either in DOME samples, or also Photorhabdus or Xenorhabdus. Maybe if someone knows better, please correct me, but I excluded them for this one. And then I left in the GNPS motifs, which are more general motifs from the GNPS libraries. Also the mass bank is more general motifs. And I also left in the urine motifs. We are not looking at urine data, but who knows, maybe there could be something in there in the DOME samples, I don't know. But yes, here this is of course very dependent what you think your data set contains. And so if you hover over those motif databases, you can see how much motifs those contain. So the free motifs, it basically this number will add up to the free motifs. So if you already have 300 motifs defined here, and you include all of those, I think each of those contains around 100 motifs as well. You would estimate that there are 500 motifs in your data set. So that's, in my experience, a bit high. So I would lower to the free, would say 100 free motifs, and then around 100 of these three motif databases. So it's about 303 motifs estimated altogether. And then there is some scores that you define. Usually the default is just fine. If you have more specific questions or want to play more with it, you can also do that on the on the MS2LDA org website. We won't go into that in this workshop, but there is a lot of material online as well that you can explore. But basically the overlap score, Justin, correct me if I'm wrong, it defines how much of the masked motif is in the spectrum, whereas the probability score would define what proportion of the spectrum is explained by the motif. And then the top X in node is how many motifs you in the end want to map onto your GNPS network. So in fact, we will, in addition to the cosine connections in your network, you will have other lines that will be the motifs. So the motifs will be directly mapped onto your network. And then here, this is Daniel's account. So it automatically populates with his email address, and it will do the same if you submit it within your account. And then we just press submit here.
So Madeleine,
Yes.
I think you explained very well. So it's just that it's good to realize that MS2LDA is a Bayesian approach. So basically everything has a probability to belong to everything, right? So sometimes these probabilities are really low. So the reason that we use some cutoffs on the probability is to get rid of all the very, well, unlikely links, basically. And the overlap score was introduced to also remove the occasions where the algorithm is, each spectrum has to belong to one motive, but sometimes that's simply not the case, and then still they get included. So basically this overlap score helps to get rid of such spurious kind of links that are not real. So you can play around with the exact value. The higher you put these things, the less populated the motifs become, but the more real links you will also throw away.
Right. Thanks, Justin. So yes, this was to the MS2LDA. We are short on time, so I will continue here with also MolNetEnhancer. How can I access the full screen here? Daniel, do you know? F5, okay. Okay, yes. Okay, thanks. Great. Also, the online people can see the slides. Yeah. Okay, we went through this, the MS2LDA job solution. Okay, yes, so now to MolNetEnhancer. So I think I didn't introduce myself properly yet. So I'm Madeleine, and I'm currently based in Copenhagen. We have a clinical metabolomics lab, and we'll be introducing a bit more about our work tomorrow as well at the stat session. But so what I'll be showing right now here is the MolNetEnhancer workflow. And this is a workflow I designed together with Justin and collaborators during my postdoc at Peter Dorstein's lab. So this is all work from the past, basically. So the overall idea of MolNetEnhancer is the following. So maybe you now, as at the workshop as well, you ran a molecular networking job. Maybe you also did some in silico structure annotation. Maybe you used, as Kai showed today, CSI Finger ID or any other in silico tool that's available out there. And also then, as we just submitted the job as well, you did some substructure discovery using MS2LDA. And you might wonder, all of these, they come in different formats, and the results are on different websites. And it's really not trivial on how you combine this huge lot of data into something easier to digest and something more easily interpretable. And so the idea of MolNetEnhancer was really that we wanted to integrate those different outputs into one network and a bit more easier, understandable and interpretable format for the end user. And so how did we do that? So basically, what we did is we collected all the structures that we found using any in silico structure tool, as well as the GNPS library matches. And we submit them to automated chemical classification. So the classifier tool that Kai also showed today. And then we created a kind of a summary on the network that will highlight which was the most predominant chemical class that was found by in silico and GNPS library hits within a molecular family. But at the same time, we then also mapped the MS2LDA motifs within the network so that we would also get some more subtle substructural differences of our chemical data. And so a bit more detail on how how this is done, or how we designed this algorithm. So basically, all those structures that you retrieve from in silico structure annotation or GNPS, they will be summarized into chemical classes to the network. So you can highlight them at different chemical class levels. And the way that it's done is through classifier. So basically, it's a tool where you submit SMILES structures, and classifier will give you these chemical classifications so there's different hierarchical levels, chemical ontology, basically, that also Kai, I think, already nicely introduced. And then so on the network, we will explore, so we get different matches, right, per mass spectra. So we have always a lot of different chemical structures per node. And MolNetEnhancer will look at those structures and classify all of them, and saying, like, some are flavonoids, so some are coumarins, and so on. And then we will calculate which would be the most predominant class in this molecular family. And here we see, so most nodes are green. So we would say 2.25 of the nodes in this molecular family out of 6 nodes are flavonoids. So the MolNetEnhancer tool would retrieve for you 37% or most molecules of this molecular family are classified as flavonoids. So I think here it's very important to see that it's just providing you a summary data. So it's by far not comparable with a very sophisticated algorithm that Kai talked about with predicting actual chemical classes, but it's more to guide your manual structural annotation and to give you a bit of a hint where you could look for. So it's not the absolute class that's true, it's just a hint, basically. And so this most predominant chemical classes, they are calculated at all levels of the classifier ontology. And what you get back is what Justin also showed a bit in his slides already, is like these colorful networks where you can highlight different classes. And as we know through library matches, we can only retrieve 2 to 5% of our data with chemical structures. And in this way, by using a tool like MolNetEnhancer, you can enhance this chemical structure annotation to about 40%, meaning not annotated, but at least you might get an idea of more broad chemical classes that are found in your data. I think very important is to highlight here that don't trust those classes. It's really just something that can guide your manual structure annotation. So these classes don't necessarily show the truth. They are just, as I showed, a summary of the most predominant chemical classes that have been retrieved through the in silico tool of your choice and through the GNPS library matches. And just to give you a bit an idea of what I mean by that and how you can interpret such a network, if you cannot trust it, how do you interpret it then? So I brought a little example that's from lab 15 from the DOME samples that we're working at. And we have here a little molecular family where with different masses, and GNPS didn't retrieve any structural hit for us. So, okay, what do we do now? Basically, we know nothing about those features. But so if we run an in silico tool, here I chose to run a network annotation propagation, which is based on NetFrag. I will say some words about that as well. But so it's just an alternative to serious CSI finger ID, basically. And what we get then is this myriad of structures. But how do we know which one is the correct structure? And even is there something that's meaningful to us? So what MolNetEnhancer can provide is, it would in this case tell us that a majority of compounds that were predicted by in silico structure annotation are organoheterocyclic compounds. So we already get an idea, okay, it's probably something, yeah, a lot of things can be organoheterocyclic compounds, but we get a bit an idea of what kind of compounds it could be. And then we can, in addition, also map the motifs. And in this case, we see that all of these structures, they share a master motif, which was annotated as a urine master motif 64. So it's a C6H9NO3 substructure. And then in our in silico structures, we can try to see, are there any structures that actually contain this substructure? And we can see that 2 of those structures actually have the C6H9NO3 substructure. So this is just how you can combine different tools to get a bit more confidence in your structural annotations. Again, as Kai said, most likely it's not the exact structure, but at least you get an idea what sort of compounds that could be. So here you could maybe conclude that those compounds could be organoheterocyclic compounds, and maybe they are tetrapeptides which contain the C6H9NO3 substructures, which would be something really typical or common in a tetrapeptide. And so this would correspond in the metabolite identification levels to a 2 to 3 level identification. Again, it's not the exact structure that you will get, but I think it's much better than really knowing nothing about the structures as per se you would by just using the GNPS network. Yes, and also MolNetEnhancer, you can run it directly within GNPS. We will, if we have time, I think we do, we will go through this as well. But before we run this, we also need to do an in silico structure prediction. And the way MolNetEnhancer is set up on GNPS right now, it accepts in silico structure input from not a serious CSI finger ID in that case, but network annotation propagation. And so before we can start MolNetEnhancer, we also need to run an in silico structure annotation through network annotation propagation. And just to say a very few words about this algorithm. So basically, network annotation propagation is based on the algorithm that's called NetFrag that Kai also mentioned, and it uses structures from databases as input and then tries to predict a mass spectrum, and those mass spectrum are then matched to your library to your experimental spectra. And so what network annotation propagation does is it then re-ranks those candidates based on the network. And it does that assuming that the library matches are sort of the ground truth, so the most correct matches, so to say. So if you imagine you have one molecular family here, the red ones would be the MSMS library matches, and the blue ones would be in silico matches. So you can imagine if you have these three structures predicted in silico, it will compare those to what you retrieve from the MSMS library matches, for example, in GNPS, and then it will re-rank your in silico structures based on the most similar structures to the GNPS library match. So in this case, it would be this hexagon that would be ranked as #1 because it's most similar to the neighboring MSMS library match. Okay. And so network annotation propagation is, you can use that through GNPS as well. And we can together now see how you will do this. Also, this is all the steps you need to do. They are on the slides that are available for you in the folder. So don't worry if you cannot follow in the pace right now. So we go again to the GNPS website. And then you scroll all the way down to in silico tools here under advanced analysis tools. Select browse tools, and then you see here network annotation propagation and a little blue here where you click on. And then it will open up the network annotation propagation job submission website. So again, you can name your job submission. We just name it Intralab DOME, and then we say NAP. And then here, the most important parameter here is, of course, your GNPS job ID. So we can go back to your jobs. And let's say you want to do NAP of this feature-based molecular networking job, then your job ID can typically be found up here in the address. So you can copy this and insert here. Yes. And then the number of a cluster index, you can either select the ID of one node from where you want to start the network annotation propagation, or you can set it to 0, which is kind of a hack. It means that it will do network annotation propagation for your whole network. So here you can leave that at 0. And then N first candidates for consensus score is just how many candidates it will take for calculating this re-ranking score. So leaving it at default is fine. Then the exact mass, of course, depends on your instrument again as well, how much ppm error you expect. Then acquisition mode is, of course, for us positive, and in most cases positive. And then here structured databases, basically you enter from which structural databases the algorithm should take the SMILE structures to predict the mass spectra. So here, if you don't have any specific idea of what structures you will find, you can just use all of the databases. So you would just type here GNPS and Coconut, HMDB, and so on. So you can, for your job, you can select all the databases. And then the rest you can pretty much leave on default. The adduct ion type, you have the choice to select different adducts. If you expect any special types of adducts from your experiments, typically I just leave it on M+H, which is the most common one. And then the cosine value to be subselect inside a cluster, you can leave it at 0.5. The important thing here is that it's lower than the cosine score you selected to create your networks, because that it will base this cosine score, it will be looking at the neighbors. So if you set it higher, it will not see the neighbors that are actually connected in your network, if that makes sense. And then again, your email, and that's it. So here we can submit. And so this typically takes a while to run. So in the meantime, I can show you how to run a MolNetEnhancer job. So for the MolNetEnhancer job, you just again go to your feature-based molecular networking job. And here you have a handy link as well again, which says enhance with MolNetEnhancer. And again, you give it a title. And then there is no parameters you need to set here. The only thing is that you need is the GNPS task ID, which it already automatically pre-populates for you. And then also the NAP ID, which is from the job we just submitted. You need to wait until it's finished, but just for the purpose of demonstration, I copy it in here. So you would copy this job ID from the NAP job and fill it in here. And then also, if you use other in silico tools that are implemented within GNPS, such as dereplicator or WarQuest, this is more for peptidic natural products, and we won't cover it in the workshop, but there's also a lot of online material if you're interested. You could also provide those job IDs here. Then the MS2LDA, the LDA job ID, unfortunately, doesn't work. So just don't use this parameter here. But you can combine the outputs manually afterwards within Cytoscape. We won't have time to look at this right now, but maybe there will be some time left afterwards when Daniel shows how you analyze your data within Cytoscape. Otherwise, there's also slides that I shared with you where you can look at step by step how you create the MolNetEnhancer integrated network. But here we can just click submit. And then just as a little teaser on how that looks like in the end. I have here the integrated network already. So what you will see then in your integrated network is your chemical classes. You can highlight different levels of chemical classes. So here it's the superclass, but you can also do the, let's say, subclass, color them differently. So you can highlight them in your network, and also if you zoom in, you can see that the different motifs are also highlighted with different colors here. So you can use this information in addition to the GNPS network to help your chemical structure annotation. Yes, and this was already the end of this part. I think we made it pretty much in time. I put some more resources and documentation for you and in the slides, and also all links to all the GNPS network shops that are already done. So if you do it on your own data, you will have to wait probably until tomorrow until those jobs are run. They take quite a long time. But if you want to explore beforehand, you can use those links that are provided here. And then also on the supplementary slides, I explain how you integrate all of those results within Cytoscape. Yeah, there will be some time for you to work on your data afterwards. And yeah, feel free to ask me anytime, or yeah, also write emails if you encounter any issues. Thanks a lot. Are there any questions right now already? Yes?
You physically can run the GNPS job with the MolNetEnhancer without the NAM.
That's true, yes.
It's like less accurate, or just don't do it, it's wrong?
I mean, you can do it if you have, let me show that slide again. So here, basically, I think because it creates a summary, it makes most sense if you have a lot of structures in your molecular family. If you only look at GNPS library hits, it's likely that for many molecular families, you either won't have no hit at all, or maybe 1 or 2 hits. And so the summary data it will provide you will be like, I don't know, 1 out of 20 nodes is this chemical class. So it's, unless this hit is super accurate and all of the nodes that are connected to it, it could make sense. But otherwise, yeah, it's the best is if you have as many structures as possible. Yes, any more questions? Yes.
Well, it comes to me later because I ran it and it gives an error. System doesn't
Okay. Okay, sure. Yes, I can come to you and we can find out. I will come to you right after. Then maybe we have some online. No, I don't think we have online questions. Yes. Yeah, there's one. No. Okay, I can have a look, and maybe it's a problem for everyone, then I will show again on the screen. So I just wanted to show 2 things that I think were the main issues when the job failed for a network annotation propagation. Yes. Sorry, forgot to share the screen for the online participants. So now that should be working. So I just go to my NAP job again. So one thing that's a bit tricky if you work with NAP is that it's run not on the GNPS interface, but on the Proteomics 2 interface. So if you actually open your job here again, you would get this notice, could not find results, use specification for workflow. But if you just change in your browser the address, you replace GNPS with Proteomics 2 and hit enter, then you will be guided to the right website. So actually one of the problems that occurs when submitting an NAP job is that here I'm already logged in right now. But if you're not logged in, you need to log in yourself again up here. And it's exactly the same username and password as for your GNPS account. It's 2 different platforms, so it requires you to log in again. Otherwise, the job won't run. Okay, so I'm logging in again. And then there was another issue that we observed, which is my mistake. I didn't specify that. But here for the workflow type, you actually need to select MZ-MINE. So it just specifies that the data format your feature-based molecular networking job is from an MZ-MINE preprocessing output. That's something the algorithm needs to know. And then everything should hopefully work. Were there any other issues that popped up that we haven't discussed maybe yet? Yes?
Maybe from this I go to the next step.
The MolNetEnhancer.
Yeah.
Yes.
From here to the next.
Yeah. So basically, here you can just submit. So this will run your network annotation propagation. So if you're working with your own data, you actually need to wait until this job is finished, which will take several hours. So probably you will have to run MolNetEnhancer on your own data tomorrow. But just to show you how you then do it. So again, you go to your to the feature-based molecular networking job. I have the link somewhere here. So this is just from our example data. Copy it. So this can be the link to your own data set that you processed yesterday on GNPS. And then you go to Enhance with MolNetEnhancer. And then here, there's very few things you need to specify. So the GNPS ID is already pre-populated. And then here you would enter the network annotation propagation ID. So the NAP job you just started to run. The ID can typically be found up here in the address. So you copy this and fill it in here. And then you basically just click submit. But yeah, you need to wait until NAP is finished. And the same is true for MS2LDA. If you want to run MS2LDA, you also go to your feature-based molecular networking job, and then you select Analyze with MS2LDA. Yes? Sorry, I had troubles. Yes. Sorry, what's the question? If it's not finished, then actually, I'm not sure. I never tried. Yes, then I think that's it for now, unless there is more questions. But feel free to ask also later if something more pops up. Then I think we can continue with the cytoscape.